In 2026, I'm sure we've all seen our fair share of true crime TV and media. In all of these shows, the concept of forensic evidence is a pillar of the plot. One of the most important kinds of forensic evidence is blood. Blood is the only bodily fluid that primarily remains inside the body at all times, so when it's found outside of the body, like at a crime scene, it is very significant. Since it is so special, it is important that we can properly identify it when it pops up. Most people are familiar with the common identification methods, like using luminol or old-school ABO blood typing, but before we get into those methods, it is important to go into the basics. What is blood? Blood circulates throughout the body to carry oxygen and nutrients to cells and remove waste in turn. Blood can be separated into two main components: plasma, which is the fluid part of the blood, and cellular components, which are the cells. Plasma makes up about 55% of the blood volume and is 92% water, 7% proteins, and 1% of other molecules like salts, sugars, fats, and vitamins. Plasma is essentially the bus that allows all of the blood to travel around the body quickly as a liquid. Forensically, the protein component of plasma is the most significant for testing. When dyes like Amido Black or Hungarian Red are applied, they stick to the proteins in plasma to give a clearer visual of any stain present. These can also be used in species testing to make sure any blood present is actually human. Cellular components are responsible for the function of blood and can be broken up into three kinds: white blood cells, red blood cells, and platelets. First, we have white blood cells. These are called leukocytes, which literally means white cell. Their main job is to fight against infection. On the blood bus, they are the knights that defend the body. These are the only kinds of cells in blood that contain DNA, as they need the information to properly do their jobs. Because of this, they are very important when it comes to finding who a blood sample came from, as they enable DNA analysis to be performed. Next is red blood cells. These are called erythrocytes, which literally means red cell. These cells do not contain a nucleus or DNA, and perform the main job of carrying oxygen throughout the body via the bloodstream. They are like the mailman on our version of the blood bus. They do this through a special protein called hemoglobin, which binds to oxygen at specific points we call heme. These red blood cells are the basis of several different types of blood tests, which you're probably the most familiar with. Whenever you see a color change test, it is this heme component that causes a chemical reaction. You will most likely see this in reagents like leukomalachite green, which turns green when you have a positive test, or phenolphthalein, which is also called Kastle-Meyer, and gives you a pink result if blood is likely to be present. Other important exams that involve hemoglobin are chemiluminescent tests like luminol, where the reaction causes a light emission like a star, or fluorescent tests like fluorescein that glow when exposed to a specific light, like a disco ball. Both of these rely on iron that is found in the hemoglobin. Overall, if there is a chemical reaction to be done, red blood cells are your best friend. Another very important component of red blood cells is the antigens that are present on their surfaces. Every type of blood, like A, B, AB, and O, has a different kind of antigen present on its surface. This is very significant when it comes to the specific antibodies that are present in your plasma. Antibodies are utilized by our immune system and released into the plasma, or the liquid part of our blood, in order to fight off foreign cells like infections and bacteria that may be present in our bodies. Think about antigens and antibodies like different sides of Velcro. In order to keep the antibodies free in the plasma in order to fight infection, they cannot be stuck to our own red blood cells. Therefore, they need to take on a different shape in order to not stick to our cells and be able to fight other infections. These are commonly utilized in ABO blood testing, also called the lattice crest assay, where they add both anti-A and anti-B antibodies into a blood cell to see which one sticks. For example, if you have type A, you will see a specific clotting that occurs only when the anti-A antibodies are added and not when anti-B are added, which is how you can differentiate what kind of blood type you find at the scene. This is very useful for eliminating or including people like suspects or victims in examinations and detective work back when we did not have DNA technology yet. Finally, we have platelets. These are called thrombocytes, which means clotting cell. These are cell fragments that allow blood to clot at the site of injuries to prevent blood loss. On the blood bus, there are construction workers that can quickly patch up holes that they see to keep everything nice and together. They do not have any forensic tests based on them, but are still very important in the body when it comes to injuries and things that require clotting. Overall, there are a lot of very unique components to the blood that allow your body to function properly with the oxygen and nutrients it needs while also fighting off infection. We also learned today about a multitude of analyses that are done regularly to identify blood when it pops up in the crime scene and which components it's associated with in the blood. I hope you learned today, and thanks for watching.